Ronald A. de Pinho. Dr. Ronald A. de Pinho fue el cuarto presidente del MD Anderson Cancer Center de la Universidad de Texas en Houston. Antes de unirse al MD Anderson, pasó 14 años en el Instituto de Cáncer Dana-Farber y de la Facultad de Medicina de Harvard-Boston. Es reconocido internacionalmente por su investigación básica y transnacional en cáncer, envejecimiento y trastornos degenerativos asociados con la edad. Han producido una serie de descubrimientos que conducen a mejores métodos de detección temprana del cáncer, una mejor atención del paciente con cáncer y un nuevo desarrollo de fármacos contra el cáncer. Demos una gran bienvenida al Dr. Ronald A. de Pinho.
Buenos días. It's very good to be back in Mexico. I've never been to Puebla. It's a beautiful city. But Mexico is a country that I've admired throughout my career. I've had many interactions with Mexico, with its leaders, governmental leaders, with NCAN and its leaders, and I want to congratulate again Dr. Menendez-Garcia for leading such an incredible institution. This is really one of the crown jewels of cancer research and cancer care in the world. And at MD Anderson, we're extremely proud to have NCAN as one of our sister institutions. And I've also admired greatly what Mexico has done for its people. It's really a leader in public health. What you've done in the area of cancer prevention really is an inspiration for many others around the world. So I want to again congratulate you on really standing with patients. And you know, these days there is much talk about governments and administration and a lot of profiling and so on. But make no mistake about it, we're united for patients, we're united for helping patients, and there's no government or administration that's ever going to get in between what we're trying to do as a research and care community to stand with patients and families and make cancer history. So it's a big honor and privilege for me to have an opportunity to talk to you and to celebrate all of your successes. So today, what I'll tell you about is work that really forms the basis for how we think about precision medicine. It'll be a basic science talk, but throughout I'll try to make points that have significant translational potential, and these are my past and current conflict. And so what we're very interested in is really trying to understand how is it that you can identify rate-limiting genetic elements in a cancer that if you were to target them, cancer maintenance would be dissolved. And there have really been two strategies that have been developed that have actually been reduced to clinical practice that involves the concept of oncogene addiction, or the term that we use, tumor maintenance, or synthetic lethality, which is an opportunity to target vulnerabilities that are generated as a result of loss of genes, loss of tumor suppressor genes. And so these are two paradigms that have led to the development of drugs, and more importantly, not only do they identify targets, but they identify patient populations in which those targets are important. And so the concept of synthetic lethality, which was first developed by Hartwell and Friend, is shown here graphically. And essentially you have two pathways that are converging on a common biological process, and the extinction of one pathway or the other is tolerated by a cell, but the loss of both leads to cell death. And of course the shining example of that in the context of cancer in humans relates to BRCA and the fact that it's particularly vulnerable for PARP inhibitors and so on. And so these are paradigms that are leading to the development of other drugs in the context of certain genetic profiles. Now, aside from these synthetic lethal interactions, we wanted to see if there are other types of cancer-specific vulnerabilities that might emerge that really are not in the tumor maintenance concept or in the synthetic lethality concept. And that idea came from this very simple observation that when you look at a tumor suppressive gene locus and you see how it's deleted, there isn't a precise deletion of only the tumor suppressor gene. What you see here is a deletion of neighboring genes as well. And many other genes can be deleted when you delete, in this case, PTEN. And these are genes that are not important for cancer per se. They're genes that are housekeeping genes, but the cell survives because there are functional redundancies of those genes elsewhere in the genome. So it gets tolerated. So then we wanted to assess whether or not these bystander events might impact and create vulnerability for the cancer cell that could be exploited therapeutically. So I'm going to tell you about first the concept of collateral lethality. So collateral lethality, which was a concept that Florian Mueller, when he was a postdoc in the lab, he's now an assistant professor at MD Anderson, he basically had the idea that if you were to delete a particular gene, it would be tolerated by a cell because there is a second redundant family member elsewhere in the genome. And of course, the normal tissues still have both genes intact. So what would happen if you were to now use an inhibitor of that second gene? The normal cells would tolerate it because it still had the first gene intact, but the cancer cell should undergo cell lethality. And so that was a concept that we had, and actually it turned out to be a very good one, and we have many examples now of this that I'll share with you here today. So we did a systematic analysis to try to identify collateral lethality targets. So we looked for genes that were consistently deleted in cancer, that were parts of multi-family member gene families that had functional redundancy, that were co-expressed across different tissues, and that when we look in yeast, so yeast typically has one gene version, whereas mammalian systems could have 2 or 3 genes that serve similar functions, redundancy. And we looked at the knockout data to look to see which genes that deleted in human cancer were cell essential in yeast. And so that then led us to a variety of different candidates that we then attempted to validate. So I'll tell you about the first example of this, which we showed a couple of years ago, and this is published, and that is the 1p36 tumor suppressor locus where enolase 1 resides. And what we noticed is that in a certain percentage of glioblastomas and liver cancers, enolase 1 was homozygously deleted along with the tumor suppressors in this locus. Enolase is a really important gene. So there's only one enolase in yeast, you knock it out, and it's cell lethal. And in the case of mammalian systems, there are 2 prominent enolases, enolase 1 and 2, that are in different places of the genome. It's the penultimate step in glycolysis, and it serves an essential role in generating energy for the cell. And so the basic idea, again, is pretty straightforward. You've deleted enolase 1 as a cancer cell. What would happen now if you were to delete or inhibit enolase 2, either genetically or pharmacologically? And so what we found was a profound decrease in proliferation in the lower left-hand corner. You see that the enolase 2 knockdown in an enolase 1 null cell had a profound impact on proliferation and viability. And then we developed inhibitors of preferential inhibitors of enolase 2. There's a transition state analog Fa inhibitor that we modified so that it had greater stability in the serum and also permeability in cells. And what you can see in the Kaplan-Meier curve is about half of the animals that were injected with glioma stem cells that kills the animals typically within 2 months. About half of the animals were cured when we provided them with this enolase 2 inhibitor. So this is a result that we was quite impressed with because we had never seen an agent do this in vivo. So the issue, though, with enolase is it's only a couple of percent of GBMs and a couple of percent of liver cancers. So we looked at it throughout the genome for other examples of collateral lethality targets that might have a higher percentage, a greater number of patients. And so we turned to the SMAD4 locus because in SMAD4, what we found is right next to SMAD4 is mitochondrial malic enzyme 2. And it turns out that if you look across many cancers, particularly pancreas cancer, which I'll tell you about today, about a third of pancreas cancers are homozygously deleted for Me2. You can see one bar there is SMAD4 deletion, the other bar is Me2 deletion. And if you look across all the cancers, so these are just the cancer cases in the United States, what you see is about 100,000 cancer cases per year are Me2 deficient. So what is Me2? So it turns out that actually there are 3 malic enzymes, and they're very important. They generate pyruvate, and they generate reducing power for the cell, NADPH. And what you have here is malic enzyme 1, which is in the cytoplasm, and that serves cytoplasmic functions. And then in the mitochondria, you have malic enzyme 2 and 3, which are very functionally related to one another. And they generate, in the mitochondria, they generate pyruvate, which feeds the TCA cycle, and then they generate NADPH for reducing potential. And so they're very important, and Me1 is cell lethal if you knock it out. Me2 or Me3, if you knock one or the other out, the redundancy between Me2 and Me3 is such that the cells do well. And so they're able to compensate for each other. And so obviously in a Me2 deficient cell, what would happen now if we were to now deplete Me3? So we generated inducible hairpins for Me3 and tested them in both Me2 intact and Me2 null cells. And I'm just showing you one piece of data. This is published last year in Nature by PJ, who did this work, PJ Day, very gifted postdoc. And what he found was that this abrogated the ability of these pancreas cancer cells to generate tumors. So you have Me2 deficient pancreas cancer cells, you knock down Me3, and you abrogate their tumorigenic potential. And the other ones are all different controls. So now what we wanted to understand is what was the actual mechanistic and cell biological impact of Me2. Could we understand it at a greater level and use that knowledge to identify other therapeutic opportunities in the context of this pathway? So first of all, consistent with what was known about the malic enzymes, what you see in the histogram in the 4th bar is a profound decrease in NADPH production. So these are mitochondrial malic enzyme 2 and 3 deficient now. And consistent with that, if you look at ROS levels go up dramatically. So that's consistent with what we know about the importance of mitochondrial malic enzymes. So high ROS states. So then what we did, and we typically do this in the lab, we do integrated analysis. Almost everything we do in the lab, because we're tumor biologists, we're trying to deconvolute the complexity of tumor biology. So we do transcriptomics, proteomics, metabolomics, we look in mice, we look in humans, we look in vitro, we look in vivo. And we do that for everything. And so what I'm going to show you here is an integration of metabolomics, proteomics, and transcriptomics. So we did an unbiased metabolic analysis of the cells where we depleted mitochondrial malic enzymes. And what we found, and this is just the highlight, is a profound decrease in branched chain amino acid utilization. And branched chain amino acids are really important because they really help generate glutamic acid. Glutamic acid, as I'll show you later, is really important for generating nucleic acids and very important for de novo nucleotide biosynthesis. So how were branched chain amino acids utilized? So what could actually be accounting for the diminished utilization? So we knew the biochemistry here that branched chain amino acid transaminase is very important in converting branched chain amino acids through this chemical reaction into the generation of glutamic acid. So we focused our attention on BCAT2 because in the transcriptomic analysis, we saw that there was greatly decreased levels of BCAT2 at the transcriptional level. And that's just shown here, where when you knock down a Me3 and a Me2 null cell, you see a very significant decrease in the expression of BCAT2. And so we then tried to understand, well, why is BCAT2 expression decreased? We looked at the promoter, because the mRNA was down. And what we found there was something very interesting. We found that there was a binding element, consensus binding element, for SREBP1. This is a very well-known transcription factor that's very important for the regulation of many different pathways and metabolism and other pathways. And what we found was that when we knocked down mitochondrial malic enzyme and we tried to chip SRBP1 off the binding element, we found that the binding element was greatly decreased in its ability to associate with SRBP1. So there was something about SRBP1 that was inhibiting its ability to bind to its cis element. And here it goes where the ROS happens. So when you have very high ROS levels in a cell, we know that AMPK levels go up. And we also knew that AMPK kinase can phosphorylate SRBP1 and inhibit its ability to bind its cis elements. So we have a theory that the ROS activates AMPK, AMPK phosphorylates and inactivates SRBP1, and that then leads to decreased BCAT2, decreased branched-chain amino acids utilization and conversion into glutamic acid. So then we wanted to really directly look at BCAT2 and SRBP1 in pancreas cancer in meat intact cells. And what we found is that when you deplete BCAT2 or SRBP1, there is a profound impairment in the proliferative potential of pancreas cancer cells. And then the key experiment was in a Me2 and Me3 null cell, if you were to overexpress BCAT2, if it's that important, you should be able to rescue the phenotype. And indeed, that's what we show here, is that you can rescue the Me2 depleted cell tumorigenic impairment by overexpressing, by enforcing the expression of BCAT2. So this provides us with really strong genetic evidence that one of the critical elements for mitochondrial malic enzyme is the regulation of BCAT2 and the utilization of branched-chain amino acids. So what do branched-chain amino acids do? Well, actually, if you see here, I'm going to just summarize very briefly. So there are three things that malic enzymes do. One is they generate pyruvate. So we use cell-permeable pyruvate, and I'm not going to show you the data, but we show that we can partially rescue the phenotype. If we use very powerful antioxidants, which really destroy the mitochondria, the high ROS levels can lead to caspase-3 mediated activation and apoptosis, we can partially rescue the phenotype. But the really key observation, and this was a novel insight into malic enzyme biochemistry, is that it's very important in generating glutamic acid, and those then generate the substrates that are needed for nucleotide biosynthesis. And indeed, what we found, if you look at the third and fourth bars, so when you have mitochondrial malic enzyme deficiency, you have a dramatic impairment in the proliferation of cells, as I've shown you previously. And in the media now, if you simply add back on the fourth column free nucleotides, that alone is able to significantly rescue the proliferative defect. So we think that the biological basis for the profound impairment of malic enzyme deficient cells is in its inability to generate a sufficient amount of nucleic acids for de novo nucleotide biosynthesis. So this is just a summary of this first part of the talk, where we showed that when you knock out mitochondrial malic enzyme, high ROS, leads ultimately to decreased BCAT2, decreased glutamic acid, decreased nucleotide biosynthesis. And then the other very important thing, which I didn't show you the data on, was that these high ROS levels, particularly in the mitochondria, lead to mitochondrial damage and caspase-3 mediated apoptosis. So that was collateral lethality. We have many examples now in the lab, and this has already inspired several dozen drug development campaigns in large pharmaceutical companies. So we think that this is going to be a concept. It's a new approach for looking at a new target space in cancer that extends beyond the traditional oncogenes and tumor suppressive genes, synthetic lethality, and so on. And so I think that this is going to be a durable way of us thinking about ways to attack cancer in the context of precision medicine. So now I'm going to tell you about another body of work that was done in the lab this past couple of years, which is a concept that we developed called synthetic essentiality. And this is the work of Dee Zhao, very gifted postdoc as well. And so we looked at the genome, and by now we've sequenced tens of thousands of genomes. We have really good data, and we could start to generate statistical significance looking at different deletions, amplifications, and so on. And what was really interesting is that we looked for genes that were sometimes deleted in cells, cancer cells, so the cells could tolerate that loss of that gene. But that gene was never deleted in the context of a specific tumor suppressor. In other words, we thought that perhaps those genes could be genes that become important only when you delete a tumor suppressor. But when the tumor suppressor is intact, you can delete the gene just fine and the cell can tolerate it. And that gave us the idea that perhaps inhibiting that gene might be well tolerated in tumor suppressor gene intact cells, but in those cells, the cancer cells that have deleted the tumor suppressor, they should have a very serious biological limitation. So that's the concept of synthetic essentiality. A gene becomes essential in the context of loss of a tumor suppressor. And if you look at the data, for example, BRCA and PARP, so we all know that in BRCA deficient cells, you treat them with PARP and they're highly sensitive. Well, if you look at the genomic data, you can see that PARP is occasionally deleted, or BRCA is occasionally deleted, but you never delete both. And so this is an example of synthetic lethality, but we wanted to see if there was these dependencies that were in a particular pathway. And so the gene that I'll tell you about is chromodomain protein 1, CHD1. Really interesting gene. It's a chromatin reader. It binds to H3K4 trimethylation sites and it turns genes on. Regulates thousands of genes. And the only thing that was known about it at the time was that it was important for maintaining ES cell pluripotency. So that was kind of interesting. So then we looked and we found that CHD1, as you see in the top part of the bar in panel A, can sometimes be deleted. But then if you look and see, and you look at relative to P10, or relative to any of the other PI3 kinase inhibitors, what you see, mutations in the pathway, you do not see CHD1 deletion. So in other words, if you're P10 deficient or mutate the PI3 kinase pathway, CHD1 does not get deleted. So is that relevant? Obviously, this is just in silico analysis. And so what we did is we looked in prostate cancer, where P10 deletion is very common. It occurs in about 40% of the human prostate cancers. It's an early event. And it leads to initiation of cancer. It leads to high-grade PIN. It has a very feeble progression phenotype, but eventually progresses to adenocarcinoma and is non-metastatic. And so what you see here in these human cell lines is that when you knock down CHD1 in a P10 deficient cell, the growth is significantly impaired. And in contrast, if you knock it down in a P10 intact cell, prostate cancer cell, you see that there is no impact on proliferative potential. But if you CRISPR P10 in this line and generate an isogenic line, you now knock down CHD1, and now the cells become very sensitive to CHD1 depletion. So this is at least some partial genetic evidence that there's a relationship here. So then, when you look at in silico data, and for the students, you hear about all these genome-wide screens and in silico analyses, and I can tell you, you can always find what you're looking for. And there are many examples of genome-scale screens, like TBK1 and STK33 and KRAS-activated lines that really did not pan out. And so what we're interested in is not only looking at these in silico and functional associations, but also extending that to biochemical insight. Why is it that CHD1 is related to P10? And also, is there in vivo genetic evidence in human cancers or in mouse models that we can generate? So that's the next thing that we did. So what we found is that when we actually had P10 overexpressed, so that's the third and fourth lanes, the top lane, the CHD1, you can see that in lanes 2 and 4, that when you introduce P10 in those P10 deficient cells, CHD1 protein becomes less. The mRNA is not affected. And then if you do AKT inhibition, you can see that that also affects the stability of CHD1. And if you overexpress GSK3 beta, which is also the PI3 kinase pathway, you also have a decreased amount of CHD1. So in some way, P10 AKT GSK3 beta axis is regulating CHD1 stability. And so what we found is that there are GSK3 phosphorylation sites in the degron of CHD1, and that degron binds to this E3 ubiquitin ligase beta TRCP, which we show by co-immunoprecipitation in that panel there. We also show that if you overexpress the E3 ubiquitin ligase, lanes 2 and 3, you see that CHD1 protein levels go down. And then we can show that there's polyubiquitination of CHD1. So the bottom line is what we found was that P10, which regulates GSK3 beta, in turn phosphorylates CHD1, which then is responsible for binding to the E3 ubiquitin ligase, polyubiquitination and degradation. So when you delete P10, you stabilize CHD1. And we showed that the knockdown of CHD1 in that setting profoundly impairs P10 deficient tumor growth in vitro and at least in cell lines and tumors in mice. And so this then prompted us to ask, well, what is CHD1 doing? It's a chromatin reader. What is it regulating? And so what we found when we did genome scale chip. So we looked for, first of all the H3K4 trimethylation marks, and then we chipped CHD1, and we looked to see which ones were physically bound by CHD1. And then we did transcriptomic as well as the chip data, and the number one pathway that came up was NF-kappa B. And this was very interesting to us because Michael Karen had shown that there is this nebulous link between P10 and NF-kappa B activation, and it was really not clear how that occurred, but it was clear that that was the case. And NF-kappa B, in fact, had been shown to be prostate cancer progression relevant. And so this was the top pathway that seemed to be regulated by CHD1. So it led us to the idea that perhaps the deletion of P10 and then the stabilization and activation of CHD1 is then upregulating NF-kappa B, which is promoting progression of prostate cancer. And so this is the working model, that P10 deletion leads to stabilization of CH2M, which binds to H3K4 trimethylation marks, and it turns on many genes that are NF-kappa B genes, and that might be important for prostate cancer progression. So we're a genetic cancer genetics lab, so we always believe in genetics. And so we wanted to fortify our model with genetic data. And so what we did is we asked, well, if this is important in vivo, then in a PTEN deficient mouse model of prostate cancer, if you now deleted CHD1, if you generated a double knockout, you should impair tumorigenesis. So first of all, this is just a knockout of CHD1 only, and there's no phenotype. And that was gratifying because the cells can tolerate loss of CHD1. However, if you look at PTEN knockout, so this is at 7 months, you see adenocarcinoma. The double knockout shows a very benign phenotype. And the Kaplan-Meier curves speak for themselves. The double knockout, animals survive quite well, whereas the PTEN knockouts eventually succumb to growing tumors and hydronephrosis. And then we take a very, very aggressive mouse model, which is the PTEN SMAD4 model that we developed. These animals develop 100% metastatic disease to lymph node as well as some to lung. And what we found is that the double knockout, even in this very virulent tumor model, was able to largely rescue these animals from progression. So now the question is, what is CHD1 actually doing from a tumor biological standpoint? So now that we have a mouse model, we can compare the PTEN versus the double knockout and ask what's different in the tumor biological setting. And the first thing we notice is that there was a profound difference in the immune microenvironment of these cells. And when we look at the transcriptomic differences, you can see here from these different arrows that there are a lot of genes that were important in immune regulation. And so indeed, when we looked at the double versus the single knockout, so you can see at the panels, the histograms at the bottom, the first panel is myeloid-derived suppressor cells. So the first histogram is P10 knockout, and the next one is the double knockout. And what you see is that there's a lot of MDSCs, which are very potent immune suppressive cells, whereas the double knockouts lose their MDSCs. And then similarly, M2 macrophages are really high in the P10 and much lower in the double knockout. And then the last panel is the CD8 killer T cells, where you see in the P10 they're very, very few. And in fact, prostate cancer is one of the most immunologically cold tumors. And what you see is a dramatic increase in the number of CD8 positive killer T cells. So then, obviously, that's a mouse experiment. Is this at all relevant to human? And in fact, when we look at TMAs and we examine those human prostate cancers with high CHD1 expression versus low, we see a very significant tracking with immune profiles. And so we see the same shift in MDSCs, myeloid suppressor cells, versus CD8 positive T cells in humans. So we think that this is also relevant to a human prostate cancer as well. So now here's where you have a really good advantage when you have cell line data, you got biochemical data like chip data and transcriptomic data, you've got in vivo tumor data where the double and single knockout, and you can bring all of that together into one lens that you then ask, well, what is CHD1 regulating that's important for the tumor biology that we see, namely tumor immune regulation. And so if you look at these Venn diagrams, you see hundreds and thousands of genes. So that's where it becomes very difficult if you just looked at the human cell line data where you knock down CHD1 and look at the transcriptome, or you look at the chip data in mouse or human, or you look at the tumor data in vivo. Very, very difficult to really figure out what are the really important targets. But when you bring all that data together, there are 13 genes that survive 1.5-fold and only 4 that survive 2-fold differences. And one of them was IL-6. And indeed, what we found is that CHD1 is able to bind to the IL-6 promoter. IL-6 is a key molecule that shapes the tumor immune microenvironment. In fact, it's a major regulator for the recruitment and activation of MDSCs, and it also helps in polarizing tissue-associated macrophages into an M2 immune suppressive phenotype, which can profoundly impair profoundly impair killer T cell activity. So we think that this is the mechanism through which CHD1 is able to execute its tumorigenic properties in the context of P10 deficiency. So in the last set of slides, I'm going to tell you about a set of data that we generated in mouse that's now translated into new clinical trials in humans. And this really builds on the pioneering work that Jim Allison did. And in this very famous picture that he shows here in one of his many articles, he talks about the importance of immunotherapy and its ability to generate durable responses, cures for patients in about a quarter of patients. And so the challenge for us in the field right now is to try to raise this tail, as he says, by combination therapies, rational combination therapies. And so mouse models of cancer give us an opportunity to begin to discern what is, how one could actually do combinations and inform a clinical hypothesis that you can then test in patients. And so I'm going to tell you about just how important myeloid suppressor cells are. In fact, in many cancers, many solid tumors, CD45 positive cells are 50% of the live cells. Most solid tumors are actually myeloid tumors. And there's cancer cells and blood vessels and stroma and so on, but myeloid cells are in many cases the major cell type in most solid tumors. So I'm going to tell you about some work that we've done, and just a couple of remaining slides here. And that is, if you want to do multi-armed clinical trials, unless you have a lot of years and a lot of money, it's very hard to do these trials. And so what Jim Horner in the lab did was he generated mice with all of these alleles that you see here. So the PB-CRE, P10 null alleles, P53, SMAD4, and reporter alleles. And so all of these alleles, and then he generated ES cells from those mice. And then he was able to inject those ES cells into blastocysts and generate, in one setting, 100 mice that were cancer prone. Now, we calculated if we had to do this with 100 cages, it would take us 5 years to generate all the mice needed for all the arms that we wanted to do, that I'll show you in the next couple of slides. So the thing that we did initially was to take compounds that differentially affected myeloid suppressor cells and T cells. And we took agents, these three here, that had been through clinical trials and failed in prostate cancer. So the dasatinib actually was known to inhibit MDSCs, but it also inhibits T cells. Whereas Cabo and BEZ does not affect T cell proliferation, but it does affect MDSCs. And so we thought that it'd be very interesting to combine these agents with immune checkpoint blockade. And you may know that single agent therapy in prostate cancer has not generated any significant responses, so maybe 6% response rate with IPI and no responses with NIVO. And now we're doing combinations, and there may be some responses there, but not at the level that you'd want, where you'd want more than the majority of patients doing well. And so here's the way that we did this experiment. We generated the chimeras. We then saw which animals developed tumors by MRI. We then castrated the mice and fed them with enzalutamide, so they developed metastatic castrate-resistant prostate cancer, and then we followed the mice by MRI. And then we enlisted them into many different arms, and then we did bi-weekly monitoring analysis and then endpoint analysis, such as tumor phenotyping, immune profiling, gene profiling, and so on. And so I'm just going to give you the bottom line data. So if you look at the control versus the next one over, which is immune checkpoint blockade, you can see a pretty feeble response with both IPI and NIVO. You get responses, but the responses are not durable and they're pretty meager, very similar to the human situation. And then you look at Cabo, BEZ, and dasatinib. No impact on the tumor, consistent with the failed clinical trials that we saw in the clinical setting with those agents. And then you look at, let's say, the very last one, which was dasatinib and immune checkpoint blockade, and there you have no effect. And remember, dasatinib impairs T cells, so you're undoing the impact on T cells. But now the two arrows on the left panel, Cabo and BEZ, we basically had very durable responses, and the majority of animals were able to survive. And this is an extremely aggressive prostate cancer model. So this was a very impressive result to us, and it indicates that the combination of agents that impact MDSCs and reactivate T cells can be a very, very powerful one. And then we have specific agents on the right side, and those specific agents, like the P110 delta inhibitor, the PI3 kinase inhibitor, or the next one that hits the beta subunit, those also had extremely impressive results, those as first and second arrow on the right-hand panel. And then the very last one is the agent that I'm most excited about, and that is a specific inhibitor of CXCR1 and 2. And that is a very important receptor for MDSCs. And so that's responsible for recruitment of MDSCs and activation of MDSCs. So I think that agent, which is now in early stage clinical trials, coupled with checkpoint blockade, I think is going to have I could predict, is going to have very significant results for men that are suffering from metastatic castrate-resistant prostate cancer. And then this is just the last slide where this is the primary tumor on the left, the lymph node metastasis, and the micrometastasis. And what you see is that for the most part, you do see an impact on the primary and lymph node and visceral metastasis with these three agents, which further supports the idea that these agents are going to be very good in the metastatic setting to impact this. And obviously we need to look at bone, which we're in the process of doing. So I think that this is just another example where the science, really making the investment in basic research, exploiting the experimental merits of animal model systems or cell culture-based systems, or the vast amount of genome data that we now have, and all the tools to do really in-depth integrated metabolomic, proteomic, transcriptomic analyses, that this is really putting the field in a position to rationally develop agents that could impact a lot of pain and suffering for our patients. So I think, I had great conversations with a lot of students last night, and I was extremely impressed with how smart, how passionate, how excited they were. I wish I were starting my field my career today as opposed to 30 years ago. So that was a really wonderful experience for me. I love students. So here we have the working hypothesis that when we think about cancer, we really have to be looking at it like we do an organ system. And to expect that we're going to target one compartment and that's going to be a miracle, answer, I think is a bit naive, and especially for solid tumors. We're going to have agents that are going to have to at least create some kind of apoptotic death that then releases antigens that could be processed by dendritic cells. Those dendritic cells then present those peptides to T cells. And then we have anergic mechanisms like CTLA-4, PD-1, and other checkpoints that are going to become activated, and they're different in different cancers. We have many different checkpoints that we're going to be dealing with. But the deactivation of those checkpoint blockades are very important in cancer. And then very important as well is this whole myeloid space. And I think that's going to be one of the hottest areas in cancer biology, really trying to understand the biology as well as the therapeutic opportunities in that space. And it's really, if you just attack the MDSCs, you don't get durable responses. In T cells, only a minority of patients will respond to just single agent checkpoint blockade therapy. And then certainly we know from a couple of decades of targeted therapies in cancer cells that while some patients are being helped and we increase their survival, we're far from curing those patients. So I think it's going to be cures are going to come from attacking this very complicated organ system, not just in primary tumors, but in the metastatic setting. So I introduced these two new concepts of collateral lethality and synthetic essentiality that is not only about identifying targets, but identifying the clinical context, the responder hypothesis in which those targets are rate limiting. And the field has evolved. And so now we think about targets, we think about science-driven clinical trials. We've got oncogene addiction, and there have certainly been terrific examples, BRAF inhibitors, HER2, EGF receptor, mutant lung cancer, etc. We've got the great example of synthetic lethality with PARP and BRCA. Synthetic essentiality, I think the jury's still out. I gave you a really nice example of CHD1, and of course a CHD1 inhibitor or IL-6 inhibitor in the context of PDA-deficient prostate cancers is something that we can think about from a clinical development standpoint. But we don't have a lot of examples of this yet, but we're going through several hundred candidates in the lab right now. Collateral lethality, I think, is going to be a durable strategy for target identification, where lots of drugs are now being developed in that space. And then we've got to make sure that we really attack the tumor microenvironment. So combinations of targeted therapies coupled with immunotherapies are going to be the way of the future. So none of this work would be possible without the highly talented and very hard-working trainees and great collaborators that we have. Florian Mueller, collateral lethality, PJ, that work in mitochondrial malic enzyme, Zim Liu and Jim Warner and Gokong with the work that I showed at the very end of the combinations, and Di Zhao with synthetic essentiality. And I want to thank you all for your attention. Thank you very much.